Hello everyone, I'm Darshana. So in this video, I thought of discussing about the clinical trials and the published literature on the Oxford-AstraZeneca vaccine, which has been already approved in the UK and the US, and so I'm in Sri Lanka, and in Sri Lanka even it has been approved and it is being prescribed and injected to the healthcare and the military personnel in Sri Lanka. And I thought of looking into the discussing about some of the literature that has been published and some of the issues that I see as a layman, because I'm not an expert in molecular biology or immunology, but I thought it would be interesting to look at some of this literature and see what's actually being tested, right? Because one reason I thought of doing this was even so in Sri Lanka there was a huge debate on on using these Ayurvedic or traditional medical approaches to treat COVID, but and usually what most people would you know suggest is that we need to have peer reviewed research and published journal articles before we can consider using any of those approaches. So that's where I thought maybe we can apply the same principle, because I didn't see much discussion on the articles. Of course, in the media, these numbers, I mean, the efficacy levels were highlighted a lot, and everyone is using these numbers. Like one is 95%, the other is 62%, 2%, and so on. But I thought of going trying to dig a bit deeper into the actual literature and to see what these numbers actually mean and what are the methodologies used and whether there are any limitations or weaknesses in the methods used, right? Okay, so let's get into the actual literature. I will share these links to this literature in the description, right? So this is the I guess the primary research paper journal article that has been published in Lancet, safety and efficacy of, so this is the vaccine name, right? And it has a lot of authors. It has a lot of authors, and so this is the study based on the study done by Oxford University and AstraZeneca. So you can see the name AstraZeneca here. So it's not it's published by the manufacturers, the pharmaceutical company itself, right? Which is not uncommon, I guess, when it comes to pharmaceutical products or vaccines. Usually the clinical trials are done and the publications are done by the manufacturer. And the one thing I want to highlight here is that this article has been published in January 9th, on January 9th, 2021. And it says it's an interim analysis, right? So the vaccine, the trials are not over actually. It's going on, because I think we have another article, analysis, which is kind of supported by this particular article as well, that which is a newspaper article that was published several days back, early February, that Switzerland has refused to approve this vaccine because claim that they don't have sufficient data. One reason for that is apparently the trials are still going on. So this is just interim analysis where they have published the results that they have while trials are going on. And the interesting thing is that it was approved by the UK regulator on think December, yeah, 30th December, right? So which is interesting because now even before the first article on the interim analysis has been published, the regulator has given approval. Now I mean, it's kind of a hasty move, I guess, which is kind of supported by what the Swiss regulator is now saying. And also the other some other countries like France, Germany and Sweden have rejected approving it for over 65-year-olds, right? So citing that there's not sufficient data to support that it is effective. I mean, the data that is there in the clinical trials are not sufficient for over 65-year-olds, which is again interesting because that's the age group that that was supposed to be most vulnerable from COVID-19. So anyway, so that's one thing. So we have to note that this is the interim analysis. The other paper that I want to highlight is this. It's more like a review paper of that original paper, and this one has been funded. I mean, partly funded by Merck and Pfizer. And in this paper, it's a bit kind of a review, and they are kind of critical of some of the methods that were used in the original paper by Oxford-AstraZeneca, which is understandable because they are kind of competitors, right? But we have a nice summary here of some of the details. Okay, so now, so the first thing that I want to highlight is the issue with dosages. So there was some controversy about this particular study. I think we can yeah, in the review paper it says that so after phase 1 results supported a 2-dose regimen, the prior protocols were amended where necessary to require two standard doses STST cohort of approximately these kind of viral particles per dose administered 28 days apart. But a subset, lower dose standard dose cohort is one of the UK trials. In one of the UK trials, inadvertently received a half dose of the vaccine, low dose as the first dose, before change in dosage quantification methodology. Additionally, the protocol amendment enabled other trial participants originally scheduled to receive a single dose to receive a booster more than 28 days after their first dose. So there's a lot to unpack there. And so as I understand, what has happened is they were originally supposed to give one cohort 2 standard doses, 28 days apart, and another cohort just one dose, right? And inadvertently, so by mistake, I mean that's quite strange, but by mistake they gave the single dose cohort only half dose, right? And by accident. So to compensate for that, they again give another standard dose to them after 28 days. So kind of they change the method along the way to compensate for this issue. Now what this means is that the study is not designed now to cater for so the data what we have here do not accompany or do not consider the single dose or single standard dose case, which is interesting because I think in Sri Lanka my understanding is that there's only one dose given of this vaccine, and I mean the clinical trial does not seem to support that, right? So the clinical trial is done for 2 doses. One cohort has a lower dose, but still there are 2 doses, right? So whether that giving out a single dose support that, whether it's a data-driven approach, I'm not really sure. That is one thing that I want to highlight, and that is actually French president came out recently. I mean, he was a bit critical about Yeah, let's see. And you have countries that have a vaccination strategy of only giving one injection. I'm not sure that it's totally serious. So that's, I think he's hinting that that issue is there. So anyway, so I think this might occur particularly in countries like Sri Lanka, where which may not have the money to buy all the doses, so they might just give one dose. I'm not really sure, but apparently in this trial they have always given two doses, right? So that's another interesting. And the more even more what's even more interesting is this Nature article, which was again based on this study. um, the interesting thing is, so actually this article highlights this kind of a peculiar result. The So the group that got the two full doses, so apparently their effectiveness, so we will talk about how this effectiveness is measured in a while, in a moment. And so that's 62%, right? But the strange thing is the group that accidentally received a half dose as their first dose, their efficacy was, much more higher, 90%. So which is counterintuitive. I mean, usually you would expect the group that received two standard doses to be more resilient against COVID-19, because if if your hypothesis is true, and if your dose calculations and all this are accurate, you would expect that the cohort that received two standard doses to have high efficacy, to demonstrate high efficacy, but it's the other way around, right? So the article is about how this kind of a perplexing thing that the scientists are not sure. Now, when something like this happens, usually, you either there's some problem with your methodology or the way you calculate the efficacy, or even more serious could be that your understanding of the vaccine might be incorrect, right? Your hypothesis might be incorrect. So, but regardless, the regulators have gone on to approve it, right? Which is again a bit interesting. And so yeah, so there was another paper that was it's a preprint. It was the preprint was published pretty recently, 1st of February, right? So it's not peer reviewed yet, and as I understand, it is also based on some of the data that was obtained from this ongoing study, and that and there was wide publicity about this preprint, that it kind of the most of the media articles claim that this study shows that the AstraZeneca vaccine cuts the transmission by 67%. Now there also, so we'll come back to that transmission issue in a moment. But what I want to highlight is this. So there's some other articles about that. So now in this particular preprint, right? Yeah, so this is kind of summarizes what's given in that paper. What they found was that while a single dose of the two-dose vaccine cut positive test results by 67%, the figure many media reports seized on two doses reduced the positive test by a lesser amount, 49.5%. So again, it's interesting because apparently when you compare the single dose with two doses, the two doses actually is appears to be less effective, which is kind of counterintuitive again when you think about a epidemic, right? I mean, as the two dose approach was the originally like uh, original approach that was that it still is supported by the manufacturer. When you give a single dose, apparently here the effectiveness is higher. So again, you have to either there's a issue with how you measure the effectiveness, and or there's an issue in the original hypothesis, which is a serious, which could be a more serious issue. Um, so right. So yeah, that is again that's the so that's one issue that what happens, these kind of counterintuitive results, right? Whether the manufacturer or the scientists who are involved can explain that. Apparently, there's no such explanation. I couldn't find any explanation for that, but regardless, the regulators have approved. So, yeah, the other thing is now in this study, if you look at this original study, so what they actually do is they actually give the two doses, and then so they would test for some of the symptoms of COVID, and they would do a swab test, something like the NEE test is kind of a broad name for PCR test, as I understand. And they would after a certain amount of time, they will maybe 14 days, 21 days, they will look at the occurrence of like positives in the PCR test and the other symptoms in the control group and the in the group that was that for which the vaccine was given, right? Now, so that means it only checks the prevalence of the symptoms, so to speak, which has been highlighted by the other manufacturers as well, by like Pfizer. They never claim that the vaccine stops the transmission. They only claim that the symptomatic relief will be there, and the PCR test might give a less positive rate of the PCR test, maybe less with the vaccinated group. Now, one issue with the PCR test is that even WHO recently gave updated their guideline on the PCR test, saying that you can't just go by the PCR test because it has this arbitrary kind of parameter called cycle threshold. So someone actually had given a nice analogy to explain what actually happens in a PCR test. Actually, it's the manufacturer, I guess. Now he's passed away, and the inventor of PCR test. So it's like when you want to check whether in a book there's a certain page, you can check the entire page, compare entire page with every page in the book, or you can take like a snippet of the page, like you can take a sentence or a paragraph or even a word or even a letter of that page and see whether that appears in the book, right? So now, based on the level of granularity that you are looking at, how much you zoom into that page. If you take just one letter, then there's 100% chance that that letter will appear in all the books, right? If you take the letter E. So if you zoom in too much, you can get 100% hit rate, right? So even with the PCR test, if this cycle threshold is like a relatively large number, I think they have given some numbers like 45 or something, or above 40 or something, it's integer, then usually you will always get very high false positive rate, right? If it's lower, then you get a lower positive rate. So it's kind of an arbitrary parameter, it seems, and and so that's kind of questionable whether it is very whether it's vulnerable for false positives. The other thing is now this whole mechanism, even in the Pfizer vaccine they have used the same similar mechanism, and they don't claim that this vaccine prevents the transmission, right? So in the review paper it says that although the efficacy was lower against asymptomatic infection in the LDSD cohort, unfortunately only 3.8% in the SDSL group, the results nonetheless provide some hope that COVID-19 vaccines might be able to interrupt some asymptomatic transmission. Although fewer data, 69 cases among 600 odd participants were available with this outcome, and more data are needed to confirm. So at the very least, there's no sufficient data to support the claim that the vaccine stops transmission. So again, I think the general understanding is that the vaccine will prevent transmission of the disease and will achieve herd immunity. That's not what it says, right? What it says is that the methodology is designed in such a way to measure the reduction in the prevalence of symptoms and the positive outcome or the negative outcome of the PCR test. So that's the other thing. It's really qualifies as a vaccine. That's the other thing that came. That's an issue that I had that I thought may be worth mentioning. Another interesting thing is now how they have actually measured this efficacy value, right? 62% or whatever the numbers. So if you look at these hospitalizations in the control, so this is the vaccinated group, this is a control group. Each consists of around 12,000 people, and if you look at these numbers, like 21 days after the first dose, and it's pretty small, right? The hospitalizations even in the control group, it's kind of negligible. So there's only a slight difference between the two. So how do you really compare them? It's virtually the difference. There is some difference, but if you look at in absolute sense, it's fairly small, right? Difference. So what they have done is they have used these a method called robust Poisson regression model, right? So apparently these models, I'm not really familiar with this one, so I will share the original article where this model is suggested. So it was published back in 2004. So apparently it's used in kind of rare binary events. Try to see you feed that distribution into this Poisson distribution, and then it kind of, as I understand, it magnifies that difference, right? So it doesn't magnify the error, it magnifies the difference. So that's mathematically speaking, that may be something that you can do, but I'm a bit skeptical whether that's in a real world sense, whether that's what we want. If you look at this from a point of view of an academic epidemic that has hampered people all around the world, whether these kind of negligible differences will actually make any difference. Now it's more interesting because if you look at the Pfizer the document, the report that Pfizer submitted to FDA and the US as a regulatory board, and they are actually the Pfizer trial, how they have calculated the efficacy. So in the media it was claimed that it's 95% efficient. So what they have done apparently is they have looked at the target group and the control group, right? And then look at how many PCR positives come up in the control group versus the target group, and so this is, we can see 9 and 169 apparently. So what they have done is so if we take the difference 169 minus 9 is sorry, it's 160 divided by 169. So that's 94% something, right? So that's the number that they have taken. So it's a more straightforward approach. Of course, it is also not flawless, but it's a more straightforward approach and we can understand it. But here in this particular trial, they've used this Poisson distribution, like fitted the results into a Poisson distribution and then tried to get the these numbers. So these numbers like 62%, 90%, all these numbers in this particular study is based on fitting the data into a Poisson distribution, which I think is why they did that was apparently I think because some of these numbers are not, if you look at the absolute numbers, they are like pretty small, right? So it's a rare event, and so it kind of begs the question whether whether you can directly compare these numbers. So when you say Pfizer vaccine is 95% effective and Oxford vaccine is 62% effective, yeah, we are talking about completely two different numbers, right? It's just, but we as average people, we just compare those numbers. In the media also they are like directly compared, like like a score of some sports event or something. But if you look at how these numbers are obtained, it's based on completely different approaches. You can't just directly compare those numbers. And the other thing is whether I feel whether they kind of use this method because they want to highlight how effective the Oxford vaccine is, rather than if they went on with the absolute values and just got the relative efficacy, would have been different, right? So that kind of begs the question whether you can like play with the numbers, play with the statistics to highlight, exaggerate the benefit of a certain drug or a vaccine. Yes, that's another thing that I wanted to highlight, and the other thing is now, so there is some criticism about this particular study not having enough elderly people, right? So the limitations include that less than 4% of the participants were older than 70, and no participant older than 55 years received the mixed dose regimen, and those with comorbidities were minority, right? With results of that subgroup not yet available, right? So the only 12.1% of those assessed for efficacy were older than 55 years, right? So it's kind of strange because I think if since COVID-19 is supposed to assume to be deadly for elderly people, why it's in the trials only relatively few elderly people were used, and that has actually amounted for some criticism, and that's why apparently all these countries, European countries, have not, and they say that we don't have sufficient data to approve it for elderly people, right? All right, so yeah, that's another thing that I want to highlight, and also now these trials have been run in different countries, and all these results are pooled, I think. So it's a pool analysis, right? So we don't see the breakdown in some breakdowns are there, but the efficacy values are based on a pool analysis. So and it's interesting that so some of these studies in some countries, they were single blind experiments where the healthcare workers or the people who were doing the experiment knew what the placebo was and what the actual vaccine was, right? And one group had a double-blind study, and the way that the positive cases were identified were also different in different groups, right? So um, yeah, so in different countries there were different ways of identifying the cases, and different ways of doing the PCR test, right? In some cases they were asked to do it by their own. I'm not sure how effective that would be, because apparently the PCR is difficult to self-test, right? So I'm not sure why there are so many differences in the test, right? So even the quantifying systems were different in different countries, right? So anyway, those results were pulled. So that's another kind of I guess something that we can highlight. I forgot to mention one thing, that is this. So the single dose AstraZeneca, this number, it says that it's based on the preprint, that the some articles claim that it shows that the transmission is cut by 67%, but apparently here also the test is based on first of all, it's not a randomized trial. That means the two groups who received the single dose may have been different, like it suggests that they were younger, and maybe female healthcare workers, right? And those who received two shots may be a different demographic, so you can't, there may be other factors contributing to this difference, right? So because this experiment was not designed to test for dosage differences, and yeah, and there are some other limitations there as well. So I will share these articles, and also the how they test for the transmission is again based on the basically the decrease in viral shedding, not transmission, right? So assuming that directly translates to the transmission is kind of questionable. So these have been raised by some other experts, right? The last point that I want to highlight is that regarding this placebo, right? So the placebo was another vaccine called MENACWI, which apparently is given for meningitis, right? Now, if you look at the adverse reactions, so the review article says that there were 175 total adverse reactions, and 85 in the vaccinated group and 91 in the control group. So immediately you kind of wonder whether how come in the control group you have more adverse reactions. That's because that's also an active vaccine, meningitis vaccine. So I think that's kind of a common practice in vaccine trials. You give the control group another vaccine. The issue there is that usually when you say a placebo, the general understanding is it's something that is not very bioactive, which is not unable to do some actual biological biochemical changes in your body. But when you give another active vaccine, so that can cause some side effects, which might underplay the actual side effects of your targeted vaccine, right? So it's like when you're marking a paper of a student based on another student's paper and not based on the marking scheme, you kind of you might give a higher mark to the student because the paper that you are comparing with may also have some mistakes. So I'm not sure whether that's the optimum way to do that. My understanding is it should be something that is not very bioactive, but maybe there's some justifications, and that's something, another interesting thing that I noted, that the placebo is not like it is a very bioactive vaccine, right. So yeah, those are the things that I want to highlight. So some of the limitations in my opinion in these trials and the publications, and I will share these details in the description. And so apparently still in different countries, different regulatory bodies have different opinions about. So in Switzerland, they have basically refused to approve the whole thing. They have claimed that the data is not sufficient. In France, Germany, Sweden, they rejected it for 65 years, and whereas in the UK, US, and even in Sri Lanka, I think Sri Lanka follows the UK standard, I guess, and sometimes the US standard. But anyway, I didn't see such restrictions. So even among different regulatory bodies, there are different opinions, it seems, based on the same data, right? Which is kind of interesting because it kind of points to some subjectivity in interpreting the data. Maybe there are some other factors. My hunch is that there may be some other factors like political pressure, and there are some there's a lot of money involved in this as well. I'm not saying that they are kind of there's a lot of money involved, and they have invested a lot of money, right, in these trials and all that. So and you have these other vaccines getting rolled out, so there's a pressure to approve these and roll it out as soon as possible, because it's a competitive thing on one hand. So yeah, so do share your thoughts about these things that I discussed, and if you have any other opinion, and if you see any other issues, so I will share these articles, research papers, and the other supporting articles in the description box. Thanks for connecting, and see you again with another video.